everyone. Thanks for having me. It's so great to be here. That was a great introduction. I guess there's nothing I really need to add. Don't worry. I will explain what all of these words mean. I will just jump right in. I'm definitely happy if anything is confusing along the way to answer any questions. This is like weird interdisciplinary research. So always happy to like do what I can to make sure we're all on the same page. So first, let me just start by explaining what eco-evolutionary dynamics are. So basically, they are the way that ecology, so ecology is the set of interactions among species in a community and also maybe species between, or interactions between species in their environment. So eco-evolutionary dynamics is the way that ecology interacts with evolution, which, as you're probably comfortable with, is kind of, this process by which species evolve to be better and better at surviving and making copies of themselves through reproduction. So there are a lot of interesting questions that you might ask about evolution and ecology each individually, but there's also a lot of questions you might ask about the ways that they interact with each other. For a long time, it was thought that kind of ecology and evolution operate on separate timescales, and so you can think of them as kind of these two separate forces. But increasingly, it's become really clear that that's not the case, that ecology is shaping evolution and evolution is shaping ecology. And so if we want to predict what's going to happen in nature or in medicine with the progression of evolution, we really need to be thinking about kind of both ecology and evolution. And so if we want to answer these questions, there's a lot of different systems where we could ask them. Most of you are probably at least somewhat familiar with the idea of field research, where you go out into nature and you collect samples, you study what's actually happening out there in the world. Most of you are probably also familiar with the idea of laboratory experiments, where you can do a much more controlled type of experiment. In evolution, we have some very high-profile laboratory experiments, such as the long-term evolution experiment, which was until very recently here at Michigan State University. That's a really cool experiment where they have been propagating 12 lines of E. coli for, over 30 years now. The experiment is slightly older than I am. And so that is an example of you can ask questions in that system that you just couldn't ask in the field because you have so much more control over it. And you can watch many generations of E. coli elapse. They've actually seen what is arguably the evolution of a new species of E. coli, and they've watched exactly how it happened. And so being in the lab gives you a lot of control that you don't have in the field. But it comes at the cost of kind of sacrificing some of the rich dynamics that you would see in nature. There's been a lot of interesting research on how when you take bacteria and stuff into the lab, you might think that there are trade-offs in their life that they don't actually experience in the world. Just because you put them in a simpler environment where they're lacking some of what they would actually normally have, like out on the leaf of a plant or something. It is necessary to switch to the lab for some experiments, but there's costs. Similarly, if we want to go one step farther, we could switch to doing digital experiments. And so the way that these work is we create lifelike systems, so basically computer programs that exhibit some of the properties of life on Earth. And we can build these systems, and we can kind of create them from the same ground rules that nature operates on. And we can watch what happens in them. We can basically treat them the same way that people treat other model organisms in biology, like, Drosophila or Arabidopsis, any kind of organism that people often do research on. They'll, ask questions about how evolution or ecology play out in that organism and try and make inferences about how that generalizes. We can do the same thing with these digital systems. And obviously, they have some fundamental differences from evolution on DNA or any evolution that we see on Earth, but they're fundamentally operating on the same principles. They work for the same reason. And so if we see phenomena happen in these systems, and we also see them happen in the lab or the field, that's really strong evidence of generality. And also, if we see things happen in digital evolution, and also we can do experiments in digital evolution that would just be impossible to do in the lab even. we can measure things that it is not possible to measure. We can manipulate things that could not be manipulated. Often we'll manipulate like the mutation rate, so the rate at which there's like errors during self-replication. And you can't do that in the lab, but it is really a necessary kind of experiment to do to ask questions. And, we can continue along this continuum where we're trading off rich natural dynamics for the ability to do fast and transparent experiments. And we'll come to kind of some systems that maybe, again, you're more familiar with, like simulations, where you're really trying to directly model a specific phenomenon, or even mathematical models. In most of the research that I do, it's just not tractable to make a mathematical model out of it, although I'm always very excited when I can kind of like back one out once we've learned enough. And there's kind of a rich space of systems that exist kind of in this simulations and digital evolution zone. So I'm just going to talk through a couple as examples. And I'm assuming that if there were any questions in the chat at this point along the lines of help, I don't understand at all, that then would pass them along to me, because I definitely want to make sure that this all is like, a comfortable level of explanation before I delve deeper. Cool. Thanks. Okay, so like I said, you're probably all comfortable with the idea of simulations. And so simulations are an attempt to really directly model some sort of a biological situation. So in my research, during my postdoc, I did a number of simulations that were really, directly targeted at replicating an experiment we did in the lab. So for instance, this one here is one where, so just to briefly explain, my postdoc was all about cancer evolution. I was working at Cleveland Clinic. And one of the things that we know about cancer evolution is that in tumors where there are regions of low oxygen, it is more likely that those cancers will evolve resistance to drugs and other treatments than it is that they'll evolve that resistance in tumors that don't have low oxygen regions. So one of the projects we were doing was to try to figure out why these low oxygen regions can lead to increased evolution of resistance. And so to test this in the lab, we had this really cool 3D printed system called a memic plate, developed by the Carmona Fontaine lab. And basically the way that it works is there's these little chambers that are almost entirely sealed off from the air, except for a little slit on one end right here. And so oxygen can get in through that slit and it will diffuse through the rest of the plate. But there are cells growing on the bottom of the plate and they're using up that oxygen. And so you end up with basically a gradient of oxygen from the slit end to the closed end. And along that gradient, you end up with, a differential level of ability to survive. And so you can kind of study how evolution proceeds at these different levels of oxygen. And so during my postdoc, I built a model that was really directly replicating that exact system, but allowing us to, again, do measurements that we couldn't do. So, I built this model that behaved the same way. And then we looked at, for instance, the phylogeny or the ancestry tree, basically. So the tree of like what was descended from what in this world, which we tried repeatedly to come up with a way to measure in the wet lab, but it's much harder to do that. And so by having this model that should theoretically be acting the same way, we could kind of start to interrogate questions about the way evolution is proceeding. I'm not going to talk a ton more about that unless people are interested. I'm happy to answer questions about it later. But that's just an example of kind of the more simulation end of this research. But I'm going to focus on the less simulation-y end because I think that is the more novel and weird and interesting end that I think might be more interesting to you all. So moving, kind of more towards that digital evolution angle, another model system that I've worked a lot with in computers is evolutionary computation. So in evolutionary computation, we're actually using evolutionary principles as a machine learning algorithm. So for instance, here we're evolving the behavior that will allow one of these little guys to walk. So the basic way that this works is it's kind of this loop that keeps going where you start out by you've got some population of things that might be solutions to a problem you're trying to solve. And at the beginning, they're going to be really bad solutions. Like, all those walking guys were falling over. They weren't very good at it, but some of them were a little bit better than others. And so you select the ones that are better, or at least some of the ones that are better, and you make copies of them. you mutate those copies, you evaluate those mutated copies, and you keep going. And this works for the same reason that evolution in nature works. You've got kind of the 3 ingredients that evolution requires, which are inheritance. So the copies are similar to their parents. You've got variation, so they're not 100% the same. They have mutations. You have differential reproductive success. So you are having some of them make copies of themselves and not others. And if you have those 3 ingredients, evolution by natural selection is going to happen. And if we make it happen on this, machine learning situation, we end up with solutions to our problem. So that's exciting. That's a useful machine learning algorithm. It's worked on a lot of really interesting problems that I'm happy to talk about if people are curious. But there's one way that it can fail, which is it can run out of diversity. So basically, you can end up with kind of a bunch of very mediocre solutions that are all the same. And if they're all the same, there's not ever a good way for the population to kind of like explore other possible solutions. And so there's a lot of evolutionary algorithms out there. And the reason for that is not entirely, but a lot of the reason for some of it is that they are trying in different ways to maintain diversity, to make sure that they're better exploring the range of possible solutions. And so diversity maintenance approaches don't always operate on the selection step, but they often do. They often operate by kind of changing who exactly you choose to reproduce, maybe not just choosing the best, but also choosing a variety of solutions. And all the ones I'm going to talk about today kind of operate at that step. And so I'm going to come back to the concept of evolutionary computation in a second. I just want for now you to know this is an example of a lifelike computer program in which we can study eco-evolutionary dynamics. And I've kind of used this as a model system for asking some questions about eco-evolutionary dynamics. For now, I just want to make sure you kind of understand the full spectrum of lifelike computer systems that are out there for asking these questions. So the far extreme of the kind of complex, rich, lifelike dynamics are the artificial life platforms. And these are really just trying to be kind of their own world inside a computer. There's a lot of them. The one I have a video of here is Evita, which is probably the most well-known. It's definitely the most well-known among evolutionary biologists. And it is a really fun system to think about. So I'm going to go to the video in a second so you can kind of see the process of life in Evita. But just to prep you, basically, Evita is a world populated by self-replicating computer programs. Sometimes we talk about them as like domesticated computer viruses. And so their genomes are literally little computer instructions, kind of like assembly code or like the very simple instructions that your computer would have all of its processes made up of. And they are very literally containing the set of instructions necessary for the organism to replicate itself. Okay, so I'll start the animation. So here we have an organism. Actually, I'll pause for a second. So this is the organism. It is kind of a virtual mini computer. So the genome is the code, but then the kind of, body you could think of it as of the organism is this little CPU. So it's got 3 little spots in memory that it can use to remember numbers. It's got 2 stacks that it can use to remember some more numbers. And it's allowed to take in input from the environment, which will just be a number. There are some numbers floating around the environment that it can take in and store in one of its little memory spots. And it can also output numbers back into the environment for reasons that will become apparent in a second. And so the life of these organisms is just looping through their code and executing it. So it executed an input instruction and it took a number from the environment and put it in its memory. Now it's executing a pop instruction, which takes an thing from its stack and puts it into another spot in memory. There are some instructions that do nothing. There's an add instruction that will add the 2 numbers in its memory and put it in a 3rd spot. There's an output instruction that will send a number back out into the environment. Evita will notice if this organism is doing something like addition and kind of be able to reward that if there's other mathematical tasks that it learns to execute. And so it'll go on, it'll keep executing the code in its genome until eventually it'll hit an instruction that says to start allocating space to another position in the world. And then it's going to loop over its genome and execute an instruction called copy that copies each of its instructions into that new spot. And so it's literally looping over its genome and copying itself into another position. And now it has replicated, and now there's another copy of that organism. Sometimes when they do this replication step, there will be mutations that are randomly introduced. And as they continue copying themselves into new positions in the world, they basically, or I'm just going to go back to that last frame so that I can talk about it. So they're basically going to populate this world. Every time that an individual makes a copy of itself in a new spot, it will be overwriting any organism that was previously there. So there's basically pressure to replicate yourself fast before you get stepped on. And so that creates differential reproductive success because the programs that can replicate themselves faster are going to make more copies before something steps on them. And so as these mutations happen, if those mutations make the programs able to copy themselves faster, they are going to copy themselves faster. And so they will be selected for. And so this is a very literal instantiation of those three principles I mentioned that make evolution work: inheritance, differential reproductive success, the variation introduced by the mutations. And so we're going to see evolution in the system for the same reason we see it in nature, for the same reason we see it in evolutionary computation. And so that was Avita. That's this system here. This is just another little static representation of it. That's just one of the many systems that we could build in a computer that would undergo evolution. We often talk about evolution as being a substrate neutral process, which means it can act on anything. So in nature, it's acted on DNA to, create us and all of life on Earth. But that same process, that same algorithm would basically work on, code like we've shown here. It's often used on neural networks, can be used to evolve equations in evolutionary computation, or these are called Markov brains. Basically, whatever representation you give to it, it can optimize to copy itself faster. Okay, so that's kind of the range of systems that I think about. And all of these kind of have a role in being able to answer questions about eco-evolutionary dynamics. And to be clear, the other systems on that continuum I showed at the beginning, like laboratory and field and mathematical systems, are also really important for answering these questions. We need people working on all of them. I focus on these ones that are in a computer, partially because I think the questions I'm asking aren't all ready to be asked in the lab or the field yet. But we definitely need people working on that too. And I'm not in any way trying to discount the importance of that work. Okay, so to make the rest of the research that I'm going to talk about make sense, I want to quickly explain what my long-term goal is. So why do I personally want to understand eco-evolutionary dynamics? Well, the main reason for me is that I really want to be able to steer evolving ecological communities to target states. Okay, so what do I mean by that and why is it important? Well, there's a number of instances where this would be a valuable thing to do. So one I already talked about is evolutionary computation. I still haven't told you why that's an ecological community. I'll get there soon. But clearly it's a situation where there is a population evolving and we want that evolution to end up in a target state, ideally a state where it has solved the problem that we're trying to solve. And so if we could understand how to get it there, we could do evolutionary computation better. Another very different system is cancer. So you may or may not be aware, but most cancer isn't composed of just one cell type. It's actually composed of a lot of different types of cells, and they're all interacting with each other. And in a lot of cases, those interactions are playing a really important role in the way that the cancer is able to spread, the way that it's able to resist treatment. And so we need to understand all those interactions and how they impact evolution to be able to ultimately guide the system to a state where it has, instead of evolving resistance to our treatments, evolved to be treatable, ideally to a state where we can kill it. So that's a nice simple target state, the state where it's dead, but it's a really important one. Another collection of evolving species that evolve on a time scale that is very relevant to us is the gut microbiome. it's becoming really clear that certain states of our gut microbiome, certain like sets of species that we could have there in certain ratios are better for us than others. And we don't totally know yet which ones we want to have there exactly, but it's becoming clear that once we know that, it would be nice to be able to kind of guide that community to that state. And because all of these organisms have such short generation times, they definitely evolve over the course of our lifetime. And so being able to steer that evolution in this complex community would be really valuable. And then lastly, the one that we are very far away from being able to do, but the one that is like nearest and dearest to my heart and like is the one that kind of motivates me is large scale ecosystems like forests, the actual kind of ecosystems you usually think about walking through. Those are going to be getting destabilized by climate change. And it'd be really, really great if we could guide the evolution of those communities to a state where they are able to survive, they're able to be stable, they're able to continue keeping us alive. Agricultural ecosystems are another instance of this that's really important. And these are systems that's very hard to do these experiments on because their generation times are pretty long. But it would be really great to have these tools in our kit by the time we need them to respond to climate change. So that's kind of the underlying motivation behind all of my research. Okay, so I said that the goal is to steer these evolving ecological communities to target states. But what exactly does that look like? There are kind of two parts to this. And so I'm mostly going to talk about the second one here, but I do want to briefly talk about, okay, what does just steering evolution in the absence of an ecological community look like? Like, how is that a thing that I think is even possible? So by steering evolution, I mean basically trying to control the course of evolution. And so the circumstance people have thought about this the most in is trying to deal with antibiotic-resistant bacteria. So, for example, let's say that these bacteria on the left here happen to be resistant to some drug. We'll call it drug A. If we know enough about the genomes and the drugs and kind of the evolutionary implications of each drug, and if we have the right set of drugs with the right set of evolutionary implications, what we might be able to do is take this population that we know is resistant to drug A and give it some other drug, drug B, and know that when we give it drug B, it will probably evolve resistance to drug B. So we probably have a population here that is resistant to this drug. But if we know enough, we might be able to select drug B such that evolving resistance to it actually makes the population less resistant to drug A. And, maybe we can give them another drug after that will have kind of a similar effect. And maybe we can ultimately end up with a population that is, again, susceptible to drug A. This is an idea called collateral sensitivity. The idea that evolving resistance to one drug might get rid of your resistance to another drug. And if we can get enough data on this and we can exploit it, we can potentially help deal with the problem of antibiotic resistance and also of chemotherapy resistance in cancer. And so how would we actually do this? What information would we need? Well, the first thing we would need to know would be the fitness landscapes. And by fitness landscapes, I mean a mapping from genotype to reproductive success. So here, this is a set of 3 fitness landscapes that a grad student that I mentored during my postdoc measured in E. coli. So this is, we have 4 different genes that confer resistance to an antibiotic. It happens to be called cefotaxime. You don't really need to worry about that. But so basically the 000 up at the top here has none of those genes. And then the 111 has all 4 of the resistance genes. And then these other combinations of zeros and ones have some of the resistance genes and not others. So each of these is a different genotype. And each genotype has a different fitness in each environment. And so these are 3 different environments representing 3 different concentrations of this antibiotic. And so basically, as we are changing the concentration of the antibiotic, we are changing kind of what paths through this landscape of genotypes are selected for and are viable. And so if we're going to be able to steer evolution, we need to basically understand kind of what fitness landscapes we have at our disposal to apply to our population. And we need to be able to come up with a sequence that's going to guide it to where we want. If we can do that, which is a big if, we definitely need to do a lot of work to get these fitness landscapes and to figure out how to sequence them. But assuming we're able to, there's another component we also need, which is we need to know basically how long do we need to leave the population in each of those environments. So I've done some work on this as well. So basically, when you put the population in a new environment, you expect it eventually to reach some sort of equilibrium set of genotypes for that environment. And so here, the red indicates kind of what the equilibrium size of each population, each genotype would be, and the blue indicates what we actually observed in a simple evolutionary program that I wrote. And you can see that as we change the environment, there's a pretty substantial lag between the equilibrium and the observed population, which means it's taking a while for our population to reach the distribution of genotypes that we would see, or that we'd expect it to have at equilibrium. And if we're trying to come up with a way to sequence these environments to force the population to go through this genotype space in the way that we want it to, we really need to be able to know with confidence that it's kind of where we think it is, that it's reached the equilibrium. And so in this paper, we were able to come up with a really cool technique that we basically kind of took from quantum physics that happens to also work in evolution. Major credit to the two senior authors on this paper, Mike Hinchesky and Jake Scott. I mostly just helped with the computational side. But basically, this technique is called is it not going to play? Okay, yeah. So this technique is called counterdiabatic driving. And so here we have the control where we're not using counterdiabatic driving. And here we have the counterdiabatic driving condition. And you can see that when we use this technique, we're able to keep the population basically constantly in equilibrium. And so we can solve this speed problem. And so using this protocol, we could potentially give a sequence of drugs to a patient and know how long to give them each drug to steer their antibiotic resistant bacteria to a state where we could kill them, assuming we have the right drugs and the right knowledge about the fitness landscape and so on. Okay, so that's the basic idea of how you would steer evolution. And I have just way oversimplified that. It is a huge field with many open questions. It is in no way solved, but we kind of have an idea of how we would solve it. Whereas the second part of this problem, we have much less of an idea of how we would solve. So the ecological communities part of this question is basically about how do we do this steering when we're also accounting for the fact that there are multiple other types of species that each species is interacting with that are all going to influence each other's interactions or each other's evolution. So what additional information would we need to steer evolution in a community context? Well, one thing we would need to know is the interactions among the species. So for instance, we can represent these as these interaction network graphs. So here the red arrows indicate harmful interactions. So here A is harming B. So B's fitness is lower when A is present than when A is absent. Whereas the blue arrows are beneficial interactions. So here B is helping C, which means C's fitness is higher if B is present than if it's absent. And the thickness of these arrows can indicate the intensity of the interactions. So A hurts B a little bit. Sorry, A hurts B a lot, B hurts A a little bit. So we need to know that. But then the worst part is we need to know the evolutionary consequences of those interactions. And this is the big open question that I've been working on. And this is a huge open question. I'm nowhere near solving this. What I'm going to do now is tell you about some of our effort that we've made towards beginning to answer this question. Okay, and so the first kind of model system that I've been evolutionary computation, because we have a lot of data because people are doing evolutionary computation for machine learning. And so it's pretty easy to then look at that data and kind of draw more ecological inferences from it. And B, because it's simpler than a lot of the more complex artificial life digital evolution systems. And so we can do more replicates faster. I've got a lot of collaborators here that I definitely want to give credit to. Okay, so what can we learn about these interaction networks and their implications for the course of evolution in evolutionary computation? Well, first thing I want to do is show you what these interaction networks look like. So these are just 4 different evolutionary algorithms. You don't need to worry about the specifics of how they work. They're just 4 different ways that you could kind of try to use evolution to evolve a solution to a computational problem. And this is actually the same population. And these interactions are drawn kind of as if it were being selected under these 4 different algorithms. And so you can see these 4 different algorithms are creating really different ecological communities. This one has a lot of beneficial interactions. This one has just a very small number of intense negative interactions. So they look like they're doing very different things, which was cool for me when I first did this, because honestly, when you read about a lot of these evolutionary algorithms, it kind of sounds like they're all just doing the same thing with different words. And so seeing that they were actually meaningfully different was just a really cool initial result to get here. Side note, just to explain why I think that looking at ecology in these systems is going to be relevant, I have also done some more mathematical work showing that they're actually mathematically identical to kind of equations for ecology in ecological theory. So in ecology, people think a lot about kind of under what circumstances can one species coexist with another species. And so basically the rules for that in a lot of ecological scenarios are the same as they are in some of these evolutionary computation scenarios. And so they really are like an instantiation of ecology in the same way that they're an instantiation of evolution. And if people are interested, I have a bunch of like slides walking through the math on how these are the same, but I'm going to gloss over that because I don't think it's the most exciting part of this. So yeah, we can look at these communities from an ecological perspective. And so this is one example of a single population under each. I generated 100 populations and looked at the networks that each of these algorithms would make. And I found that they're pretty consistent in like the way that they each look. Like eco-EA kind of always makes a bunch of beneficial interactions. Lexical selection kind of always makes a small number of harmful interactions. So that's interesting. So what are the evolutionary implications of that? That was my big question, right? Well, that is a really hard question to get at. And so we're still working on it. But one of the things that we've learned so far is that we can use phylogeny as a tool for measuring this. And so by phylogeny, I again mean kind of that tree of ancestry of what descended from what. And so that's basically a tree structure. And if we want to understand the way that evolution proceeded, we can look at the topology of that tree. There's kind of 3 classes of metrics of phylogenetic topology that people use to kind of get at how evolution is happening in a different environment. So some of them will look at kind of the richness, so the total amount of evolutionary history being represented by that tree. Some will look at divergence, so kind of how far away in evolutionary space are these different species. And some look at regularity, so how consistent do the different parts of the tree look. And it's less clear what regularity tells us, but there's a lot of work on it. It feels like it tells us something. So we've been looking at these to try to understand kind of the long-term implications of these different approaches. And so for instance, here, this is phylogenetic diversity, which is a metric of phylogenetic richness. And I measured it for those 4 different selection schemes that I was showing the communities of before on 3 different landscapes. These landscapes are each just different computational problems. So for instance, this first one here is we're involving computer programs and they need to take in a number and output it square. And you can see that like these different approaches maintain very different amounts of phylogenetic diversity. And that was interesting. That was a thing I wanted to explore more. And actually, there's some preliminary data that we still haven't like cleaned up into a nice graph yet that kind of has this same story, but over a lot more different conditions and kind of more different types of landscape. And it's very consistent. Like there are very much certain evolutionary algorithms that maintain different amounts of phylogenetic diversity. So for instance, lexicase, that one that had a small number of very intense negative interactions, tends to maintain a lot of phylogenetic diversity. And so that's interesting. That suggests that, kind of that competition that it's creating is basically, allowing the different parts of the tree to coexist as they're evolving. And we're still kind of trying to figure out the implications of that. One thing that we did want to kind of check quickly to make sure that like the people in the field of evolutionary computation we care about this is like whether phylogenetic diversity actually tells us anything useful about, whether we're going to reach that target state, whether we're going to actually solve the problem. And we found that it does. So there's a lot going on in this graph. I'm happy to come back to it if people have questions. But the gist of it is basically higher boxes here mean that phylogenetic diversity. Okay, so sorry, there are two types of diversity here. So the cyan boxes are phylogenetic diversity, and then the red ones are phenotypic diversity, which is basically a diversity metric that doesn't include that evolutionary history. And the higher they are, the more information about that value early in a run of evolution tells us about fitness. So basically the quality of the solution we're evolving later on in the run. And so phylogenetic diversity is able to be a really good predictor, or at least better than other predictors, of eventual success of a run of evolutionary computation. And that's even after accounting for a number of things. So that's very exciting. That suggests that this is actually a meaningful thing to be studying. And so this line of research is ongoing. We definitely have not answered this question yet, but I think we've got a lot of indications that it's a worthwhile direction to be going, and that it can start to tell us something about the interaction networks and what their implications for long-term evolution are. Side note, we've also kind of done some research going back in the other direction, asking like, okay, so what can these different evolutionary algorithms tell us about biology? And so in biology, it's pretty common for wet lab researchers to want to evolve bacteria or something that can do a specific task or that have a specific trait. And usually when they want to do that, they do so by just kind of constantly choosing the bacteria that are best at that trait. And that works okay, but not always. Sometimes it fails. And that's actually an algorithm that people in evolutionary computation have long known is not the best way to select individuals if you want to solve a problem. And so in this paper, we actually tried kind of going back the other direction and taking some of the evolutionary computation selection methods and applying them to a simulated biology system. And it suggested that that could potentially work, that actually if you're a wet lab biologist and you're trying to evolve bacteria that can, digest plastic better or something, you should maybe actually be thinking about some of these selection algorithms. So that was a really fun little side project. I was excited that it worked. Okay, I think based on time, I'm going to tell you about one other kind of angle of attack that we've been taking on this question, and then I will end for questions. I'm never sure like how fast I'm going to go through this. Okay, so I've been talking about trying to predict the implications of a whole ecological interaction network for evolution. That is a hard problem. No one has solved it. It's also potentially useful to try and figure out what are the implications of much simpler sets of interactions for evolution. So what if, kind of a group of things are all interacting in the same way, for instance? How is that going to affect their evolution? And so we've been thinking about that in a more artificial life system. And so specifically in this work, we've been thinking about host-endosymbiont mutualisms. And so that is kind of, so a host-endosymbiont system is a system where you've got one species living inside another species. And so the species that is living, the small species is the endosymbiont, and the species that is living inside is the host. And these can exist anywhere along the continuum from parasitism to mutualism. But here we're particularly interested in what scenarios allow for that system to evolve to be mutualistic. And so that's, single pair of interactions that we're asking about. Okay, how can we determine how that is going to evolve? And so mutualism specifically is going to be a situation where both partners, both the host and the endosymbiont, are benefiting each other from this interaction. Whereas parasitism would be if they were being antagonistic and like the endosymbiont were stealing resources from the host or something. And so there's been a fair amount of research that's been done on host-endosymbiont mutualism. The thing is, pretty much all of it has been done on pairwise host-endosymbiont mutualism, where you've just got one endosymbiont living inside a single host. And this is not how systems in the real world are. You know, we don't have one bacterium living in our guts. We have millions of them. And so kind of the new question that we are trying to ask with this system to kind of, start bringing it closer to what we're doing with those complex networks is what happens when you have a lot of endosymbionts that are all co-evolving with your host. So what factors are going to promote evolutionarily stable mutualisms in that context? And by evolutionarily stable, I mean mutualisms that, aren't just going to evolve away, like where it's not going to be advantageous for one party to start cheating and then for all of the cooperation to break down. And so to kind of understand that, it makes sense to think about what dynamics can stabilize cooperation and mutualisms in the systems that we've studied before. And so I said we haven't studied really systems where we have a bunch of endosymbionts inside a single host. But what we have studied is kind of cooperation among a group of peer organisms. So like all of the endosymbionts within one host would kind of be peers. They're all on an equal playing field. And we've studied kind of this pairwise, single endosymbiont inside a host scenario. And what we found in that situation is that there are some commonalities in kind of what factors can stabilize cooperation and mutualisms. So among peers, one thing that's really known to stabilize cooperation is kin selection. So it's advantageous to expend resources to help your kin because your kin are going to be related to you. They're going to carry your genes. They're not probably going to have mutated enough to start cheating and like stealing your resources without helping your community. And so kin selection can stabilize cooperation among peers. In a host endosymbiont system, you can't really have kin because the host and the endosymbiont are different species, so they're very much not kin. But you can have something kind of similar in that you can have vertical transmission. And so that is when the offspring of the endosymbiont are very likely to end up in the offspring of the host. And so you have kind of entangled evolutionary fates. This is like us and our mitochondria. We have basically 100% vertical transmission rate with our mitochondria. And so they're very much not incentivized to try to steal resources from us or in any way not help us because they want us to succeed because the only way they replicate is if we succeed. So those are both kind of, about shared evolutionary fate. It makes sense that those could both stabilize cooperation in these different systems. Another phenomenon that can stabilize cooperation in both of these systems is division of labor. So if both partners are contributing something to the mutualism or the cooperation that the other one cannot contribute, and that the other one cannot easily evolve to contribute, then they're very much incentivized to keep helping each other because they're both benefiting in a way that they could not help themselves. So again, that makes sense. That leads to nice stable cooperations and mutualisms. Spatial structure is another thing that is known to stabilize cooperation among peers. So spatial structure means basically if you live in a spatial world. So when you reproduce, your offspring ends up near you. You're mostly interacting with a local set of other individuals that are near you. This creates repeated interactions. It lets you kind of build up knowledge of who is cooperating with who. It lets you end up again with kin near you. All of those things can help stabilize cooperation. And there's a really large literature on that of like game theorists in the 80s thinking the evolution of cooperation is impossible. And then they add spatial structure and they realize actually it is. So that's a really well-known result, which everyone, I think, or at least I certainly assumed would also work in a host endosymbiont context, but it turns out it doesn't. So this is a fascinating paper by Anya Vostenar, where she showed that there are a lot of contexts where spatial structure actually not only doesn't kind of promote host-symbiont mutualism, it can even inhibit it in some circumstances. And so the conclusion here is basically that, while we do know about some dynamics that can stabilize both cooperation and mutualisms, it's not consistent. It's not like obvious which things about cooperation among peers we can translate into mutualism between hosts and endosymbionts. And so when these situations are occurring at the same time, when we have a bunch of endosymbionts as a peer community inside a host, we don't know what's going to happen. And so what we wanted to do with this project is to kind of start laying some of the groundwork for what's going to happen in that circumstance. And so again, we care about this because that's actually the situation that most host endosymbiont systems are in. And so to study this, we are again using a digital evolution approach. There are a lot of digital evolution systems that we could ask this in. There's a really great review here, again, by Anya Vostenar. We chose to use simulation, which is kind of designed to answer these questions. So simulation is, actually, it was originally an offshoot from Evita, the artificial life system I talked about before, but kind of trying to really focus in on making the host endosymbiont coevolution as simple as possible, because it is already such a complex system that taking out some of the complexity of Evita was really helpful for understanding it. So basically, simulation, it's a pun because it's a simulation with symbionts. It's basically populated by two kinds of organisms, hosts and endosymbionts, and each one has a genome that is a single number that goes from -1 to 1. And if it's between 0 and 1, it is interacting favorably towards its partner. So hosts with positive interaction values are donating some of their resources to their symbiont. Symbionts with positive interaction values are donating some of their resources back to the host after multiplying them by a synergy factor that kind of models a very simple division of labor. So everyone can benefit here. But if the symbiont has a negative interaction value, that means it is trying to steal resources from the host. And if the host has a negative interaction value, it is trying to defend against the symbiont stealing its resources. And so it's trying to keep it as simple as possible, but even in this system, we get very complex behavior. And so previously, kind of some nice groundwork has been done in this system on how vertical transmission, so again, that probability that the symbiont's offspring end up in the host's offspring, affects the evolution of mutualism. And so here, the y-axis is that interaction value, that is the thing that's evolving. And then the pink boxes are the symbionts and the brown boxes are the hosts. And you can see even once you have even a small amount of vertical transmission rate, they tend to be mutualistic. They tend to have interaction values above 0 because it's beneficial for them to help each other. So our first question was, okay, what if we just take that known understood relationship and we add multiple symbionts to each host? Well, here it is with 10 symbionts, and you can see already you need a higher vertical transmission rate than you needed before to get the same level of cooperation, the same level of interaction values. And as we add more symbionts, this just gets worse. And basically the cooperation between the host and the endosymbiont breaks down. I should note, I was showing you averages there. There is often kind of a distribution of different genotypes and phenotypes in the population. So for instance, this is basically you can think of these area plots as being a bunch of histograms over time of the proportion of different phenotypes. And so you can see there's actually always like a very small number in this condition. I think this is like one of the intermediate symbiont count values. You can see there's always like a little undercurrent of mutualism, but it's just swamped by all these other parasites. Okay, so we see that having more endosymbionts tends to make it necessary to have a higher vertical transmission rate to see kind of stable mutualism. And thinking about it, we thought that kind of the reason for this might be that these symbionts are basically implicitly engaging in kind of a public goods game. So a public goods game is one where there's like a shared resource that helps everyone. that everyone could contribute to, but everyone could also kind of steal from without contributing. And so the resources from the host are distributed to everyone, even if some of the symbionts are actually being parasitic towards the host. And so it's very advantageous to become parasitic in that context, because you both get the resources the host is giving to you and everyone, and you can also steal resources from the host. And so we thought, okay, maybe a good first step towards understanding kind of the specifics of this would be to make that intersymbiont ecology more explicit, to add an explicit public goods game so that we can manipulate kind of the parameters and stuff and see how those affect what happens evolutionarily. So we added a public goods game. And so we added one other number to the genotype of the symbionts. It's a number between 0 and 1, and it says what proportion of their resources are they going to donate to a communal pool. And then the resources in that pool get multiplied by a synergy factor and then equally divided among all of the symbionts in the host. This is a nice little picture of how that works. And so this is also research that is ongoing. We're still learning more, but the punchline so far is that the host symbiont mutualism is stable as long as there is ongoing cooperation amongst the symbionts, which is consistent with that theory that kind of implicitly this is a public good situation. And so if we add incentive for cooperation by making like the incentive for cooperation via the explicit public goods game stronger, then we can kind of maintain that cooperation among the hosts, and that can maintain the mutualism between the hosts and the symbionts. So on the left here, we have the donation rate for the symbiont public goods game. And on the right here, we have the interaction value between the symbiont and the host. And so you can see what we tend to see in these scenarios is there's cooperation, everyone's happy, and then at some point that breaks down and they are invaded by cheaters, they stop cooperating, and shortly after the symbionts stop cooperating with each other, they also stop being mutualistic towards the host. We're still kind of digging into why exactly we see this like sudden invasion of cheaters, but this is a very reputable observation. This happens in basically all of our conditions except for the controls. So it very much seems like kind of for the mutualism to be stable in these large number of symbiont circumstances, the symbionts need to be cooperating with each other. And so we can conclude that yes, intersymbiont cooperation does stabilize kind of the collective mutualism between the hosts and the symbionts. And so this is a much simpler type of set of ecological interactions than we had in the evolutionary computation scenario. But it's one where, because it's simpler, we can start to get a stronger handle on what its implications for evolution are. And so that's a very satisfying project to be doing in parallel with the evolutionary computation one, where there is still so much unknown. So yeah, the take-home message is that if we add endosymbionts to a mutualism, we can destabilize it. But if we tweak the way the symbionts interact with each other, we can completely change the dynamics of the whole system. And yeah, we've also done some other side research that I think I'm not going to talk about because I know we're getting a little close to an hour and I want there to be time for questions. If people have questions about this, though, I can come back and talk about it. I'm just going to skip this whole last section. I wasn't sure how much time I would have. So yeah, I'm going to stop there. So thank you so much for listening. I just want to thank all of my students and my collaborators and my funding groups. And if anyone wants links to more information, they are all here. Thank you so much for listening, and I would love to take questions.
Fantastic. That was a very interesting talk, and we do have a number of questions, so I'm going to go ahead and jump straight into them.
Fantastic.
Let's see. The first one here is, how do you know if a model is over-parameterized, or is that even possible?
That's a fantastic question. So that is definitely possible with the kind of more simulation style computer programs that I work with.
Mm-hmm.
So yeah, if you're trying to exactly replicate a biological phenomenon that you're observing in the lab or the field, then yes, if you add lots of parameters, you could kind of be forcing it to fit what you observed in a way that is not actually replicating the underlying dynamics that led to that.
Okay.
And so that's definitely one of the things that stresses me out about working with simulations.
Sure.
Because when we move to these more digital evolution, artificial life models, it's harder. There's not really such a thing as over parameterization, because the parameters aren't trying to model anything that exists in nature. They're really just trying to be their own system that does its own thing. And we're going to see whether it behaves in a way that is compatible with what we observe in nature.
Okay. Okay, that makes sense. Let's see. Do simulations like Avita ever include sort of fatal mutations, like mutations that will, instead of allowing it to propagate, kill it off?
Oh, yeah, great question. Yes. Lots. um,
Okay.
so, the organisms in Avita, they have to literally contain the code to make copies of themselves. So most mutations to kind of that loop where they're replicating themselves will be fatal. And people have done a
Okay.
lot of interesting mapping of kind of like what is the fitness effect of like mutations at different parts of the genome in those systems. And there's often, just like a huge chunk that are fatal, which is also consistent with kind of what we see in DNA-based
Sure.
systems.
Yeah. Okay. No, that's interesting. Let's see. For the large scale models you talked about, so like the forest, for instance, is the limitation the number of things that need to be simulated at once, or the sophistication of the models themselves?
So I haven't actually seen someone try to make a model of evolution in a forest. I think the reason for that is that that would be trying to make a simulation where you're directly predicting what's going to happen.
Mm-hmm.
And we just don't know enough yet about eco-evolutionary dynamics or the fitness landscape of all the species in the forest to be able to do that. And so, yeah, I think it's a combination of not having enough data to
Sure.
parameterize it effectively, and then also it being you would actually need to parameterize it, and so you could have too many parameters. And also it would be very hard to know like if it were right, because a forest just changes so slowly.
Yeah, that makes sense. Yeah, I guess it would be hard to simulate a long enough time scale with everything in the forest is going to be on vastly different magnitudes. So that makes a lot of sense too.
Exactly, yeah, so that's why I say we're a really long way away from being able to do
Yeah,
that.
absolutely. Let's see. Thinking of the cancer context, is the computational model able to look at more than just genome level changes? For example, epigenetic changes or differential transcript usage that might produce different proteins with the same underlying genetic context.
Fantastic question. So I have not personally done that. I almost did that during my postdoc.
Okay.
There's definitely an idea floating around my postdoc lab that I don't know if anyone has done yet, but about, what would it look like to kind of combine epigenetic with genetic mutations? Because it does seem like epigenetic change is very important in the context of cancer. And basically, the way that we came up with how you would do that is you would kind of like incorporate multiple timescales of mutation that are allowed to happen.
Mm-hmm.
Um, Although with the epigenetic, you need to like think carefully about how to handle the epigenetic mutations there, because they're on a faster timescale, but also there may be like they are more able to revert than genetic mutations are.
Okay.
And so that's like also a factor to consider.
Okay. No, that makes sense. I can see where the complexity would ramp up very quickly.
Mm-hmm.
Let's see. Would you consider anglerfish sexual parasitism as a form of one-to-one host endosymbiont?
Oh, fantastic question. Um, so I think I wouldn't exactly call that like a host-endosymbiont system in the classical sense, because
Okay.
they're actually the same species, and like so their genetic material is shared, and there isn't as much ability for there to be conflict. I mean, there's still a fair amount of ability for there to be conflict. There's really interesting research on kind of the evolution of conflict between chromosomes, especially X and Y chromosomes.
Mm-hmm.
So like that could absolutely be a factor in that system. But it would play out in a very different way, I think, than like host endosymbiont evolution would,
Okay.
because they go through this phase of like needing to work together to produce offspring.
Oh, okay. That makes sense. Okay. Let's see. Given I think you kind of touched on this, but I'll go ahead and ask the question. So maybe you can expand on it a little bit. Given that multiple endosymbionts seem to ultimately always produce negative interactions, why do systems like the gut bacteria work?
Fantastic question. So yeah, I think a big part of it is probably that there are a lot of other effects in play yet that I have not yet added to the research we're doing in simulation. Like we've been trying to keep it very simple and kind of build it from the ground up.
Okay.
And I'm sure there's other factors happening
Mm-hmm.
that are important. So I mean, one is there probably is cooperation amongst many of our gut microbiota.
Yeah,
I'm
yeah.
sure that is happening, but also I'm sure there's just a lot of things that I haven't incorporated into this model at all. Like there's multiple levels of interaction. Like a lot of our endosymbiotic gut bacteria also have endosymbiotic phage, so viruses that infect bacteria that live inside them, and those can evolve to be mutualistic or not with the bacteria.
Okay.
And there's just so many levels.
Okay, so it just goes down spiraling. Okay, I see.
Exactly. And yeah, we're just beginning to unravel that.
Yeah. Oh, that's okay. See, now that's interesting because I wouldn't have even thought about the bacteria having their own sort of interactions going on within themselves. That's a really good point. And I could see again where the complexity would go off the charts very fast.
Mm-hmm.
And so the last thing here is okay. So this wasn't a question, but one of the people in chat, Risanabe, found a link that they think might have some examples of the one-to-one systems that you were talking about. So I'll send you that link after the stream. But
Awesome.
appreciate
Thanks.
them. Yeah, I appreciate you finding that. Hopefully it will be helpful.
Yeah.
But I think that's all the questions we have. So I'll say thank you again, Dr. Dolson. Like I said, really interesting talk. Super cool to see how you're taking these really complex evolutionary processes and starting to unravel them computationally. That's really neat. You did a great job explaining it as well. So thank you again for that.
Awesome.
Um,
Yeah. Thanks for having me and for listening.
yeah, absolutely. So if I can get you to hang around for just 1 minute, we'll talk very briefly. And for everybody in chat, we're gonna do the friendly thing and raid another stream real quick. Did I spell that right? Yep, that. So hang around for the raid and be nice and go say hi. And other than that, thanks for coming out and be back. I think next week. Not 100% on that, but we'll be back with another talk next week. And thank you again to Dr. Dolson. Have a good weekend, everybody.